So can Nairobi hear me?
Okay, please respond if you can hear me. Hello?
Hello?
Okay, that's Robert. Craig, can you hear me?
Yes, I can hear you.
Okay, good. Okay. I'll be waiting the next few minutes to see if we still have more participants joining. Then I will introduce the speaker, and then we can start off. Okay?
That sounds fine.
All right.
Okay, thank you. So we start at...
Mercy, then also give some suggestions on how to attend the seminar, like mute yourself while you're not speaking, et cetera, et cetera.
Okay, noted. Okay, so I think we are good to go, and I hope when we start off, more people are going to join us, and I think it's best we start at 10 minutes past 4. So we've got 3 minutes to start off.
Sounds good to me.
Okay, thank you. We have so many issues. So Becca has joined us for this, and from the list of participants, I think we have so many other participants from US and from UK and from Africa as well.
That's great. It's I'm really surprised that people joined us. Thank you. Thank you.
You know, it's a topic of interest to many communities of practice, and I think it will have a wide range of audience, starting from breeders to lab-based molecular breeders to private companies. I'll be sending you the list of the participants.
That would be great. I see some old friends on this list.
So to the participants, I am going to introduce the speaker of today, and I need to start by welcoming you all, and thank you for choosing to connect to this webinar presentation. this is the Genomic Tools for Sweet Potato project, in which we are organizing a series of webinars, this being the first one, to inform and educate the sweet potato breeders in sub-Saharan Africa and global in the world, and also to educate other communities of practice who are also working on crops or plants which have complex genetics like the sweet potato. Our speaker for today will be Craig Yenjo, who is a distinguished professor and a program leader in sweet potato and potato breeding and genetics program in North Carolina State University, and is also the lead PI of this project, the Genomic Tools for Sweet Potato project, which is funded by Bill and Melinda Gates Foundation. Our assistant speaker will be Dr. Mark Gisling, and who is joining us from wherever he is. And Mark Gisling, he's the Genomics and Biotechnology Global Program Leader. And he's based here at SIP Nairobi. His research focus covers the use and characterization of potato and sweet potato, word relatives, genetic mapping of traits of interest, isolation of genes and alleles, among others. So welcome all. Would request that you type your question. You type your question and we'll receive it at here Nairobi. We have 30 minutes for Craig to do the presentation, and then the 20 minutes or 25 minutes, 20 minutes for addressing the questions. So thank you and welcome all. Thanks, Craig. Back to you.
Thank you. Well, thank you very much, Mark, for the introduction. And good morning, everyone. From Raleigh, North Carolina. It's a beautiful, crystal clear spring day here, and I hope that wherever you're tuning in from, that it's likewise a good day there. I realize that some of you are ending your day. So I'll jump right into things. I don't do webinars very often, so this is a bit of a learning curve for me. And if you have questions or if I need to slow up, I'll assume somebody will type in a comment to me and send it to me. I'll be able to see it on my screen. Okay. So my title for today is an overview of what we call the GT4SP project. Our overall goals are improved breeding technologies and increased genetic gains for sweet potato. Oh, let me see if I need to get, oh, there we go. I'll try and go slow. I am continually impressed with the range of diversity that we see in sweet potato and the range of crops create from this remarkable crop. Mercy, I'm getting a lot of feedback from somebody. If people could mute, I won't have the echo then coming in on my phone. Of course, we all know that global sweet potatoes are a very important food crop globally, and this is just a picture that I pulled from FAO recently showing the global distribution of sweet potato and the major production countries. And of course, what you can see is that China is a major producer of sweet potatoes in the Southeast Asia region. And sweet potatoes are important all across Africa here, but it really is not so apparent from this figure. And then, of course, sweet potatoes in North America are actually a fairly minor crop. But I happen to live in a state where they're very, very important for our economy. I pulled this map from the International Potato Center several years ago, and it shows areas of cultivation and average yield of sweet potato across the globe. And what you'll first notice here is that in terms of yield, average yield in say China, the major production regions, are actually quite high compared to the average yield across sub-Saharan Africa for sweet potato. And our goal is to try and change this situation right here. And I do believe that if we are able to change this, then you're going to see that sub-Saharan Africa is actually going to be the major producer of sweet potatoes across the globe. And I think we can do that in less than 10 years' time. I like to put a frame of reference for my audiences generally as to what my situation is like here in North Carolina and the United States with reference to sweet potato. And in fact, we have quite a fascinating story to tell. I started working with sweet potato back in 1997, so it's a little bit off the graph. And during that short period of time that I've been working in sweet potato, we have seen acres harvested of sweet potato increase twofold. We've seen total production of sweet potato during that same period of time increase roughly threefold. And we've seen that yields, average yields of sweet potato across the United States, and particularly in North Carolina, have gone up significantly during that course of time also. Now, I think many of you know that I'm a sweet potato breeder, and the focus of my program is largely developing new varieties. And we've been very fortunate. In 2005, we released a very popular variety called Covington. It probably occupies roughly 40% of the total U.S. crop right now, and probably about 80 to 90% of North Carolina's crop. And you can see that upon releasing Covington's actually benefited through the tremendous growth in our industry during the course of the years. I would love to say that Covington was the driver of this growth, but that's simply not true. Actually, a key driver of growth in the U.S. market has really been two factors, actually three factors. One, we can store the crop all year round, and that means we can provide a continuous supply of sweet potatoes to various market sectors at any time of the year. And this has been a real enabler in this tremendous growth that we've observed. Now, here I'm showing you just a kind of a sampling of the range of sweet potato products that are currently being made from sweet potato. But truth be told, the main drivers of sweet potato product expansion right now are actually the production of sweet potato French fries. In Africa, we would call them chips, and also sweet potato chips, we would call them here. You might call them crisp in the UK and Africa also. So these two products have been huge drivers of the increase in sweet potato production, the demand for sweet potatoes, because these two market sectors have really been growing, but we're also producing juices, other novel food products, even dog chews, and even beers out of sweet potato. And I've shown some distilled products. We even make a vodka out of sweet potato nowadays. So this is one reason why our acreage has increased and our per capita consumption has increased. I'd like to say that also we're doing the same thing with in Africa too. The crop is growing in importance, and there are also a lot of new value-added products that are being developed from sweet potato across the continent. And this is just one example that Karimi sent with me, shared with me at a meeting that we had in Rwanda just last fall. These would be these golden power biscuits and fried donuts, or mandazi, made from sweet potato puree. So if you recall the same graphic that I showed you for the U.S., these are the same production trends. These are the production trends for sub-Saharan Africa. And what I'll point out to you is there's a lot of good things happening in sweet potato across the continent also. One, the harvested hectares of sweet potato is increasing. Two, the production is increasing also. But something that's really different compared to our more developed system here in the U.S. is this yield line right here. Essentially, since the early 1990s, yield trends across the sub-Saharan Africa have been fairly flat. And if we're going to improve production in sub-Saharan Africa and continue to increase the profile of the crop, we need to bend this production curve upward and increase per acre yield. And partly that's the whole reason for the development of the genomic tools for sweet potato improvement project. So our goal again is to change this equation, change these brown and red marks into yellows and greens, and thereby improving total production and creating great other opportunities for sweet potatoes across the continent. There's been a lot of good that has been done with sweet potato during the last decade, and maybe decade and a half or so. One project that I'd like to mention is called the SASHA project, or it's called the Sweet Potato Action for Security and Health. It's actually funded by the Bill and Melinda Gates Foundation. I was very fortunate to be an advisor to the foundation on this project in a technical capacity. This project was led by the International Potato Center, and if you're seeing the picture on the screen right now, I'm sure you'll recognize a number of the players there. It's made major progress in 4 key areas. One, in developing improved populations and in developing new varieties. They're making good progress in seed systems. Delivery systems have been explored, some proof of concept market delivery systems. And the SASHA project really has been very successful in developing a series of sweet potato support platforms around the continent of sub-Saharan Africa in Uganda, Mozambique, and Ghana. And the program leaders there would be the lead for the Uganda program would be Dr. Robert Mwanga right here, Dr. Maria Andrade down in Mozambique, and Dr. Ted Carey over in Kumasi in Ghana, and of course Jan Lowe, the overall project leader based in Kenya. I'm not sure who's all attending the webinar today, but if you're here and I just mentioned your name, I know Robert is here. A quick shout out to you guys. You've done great work and continue to do so in the future, I'm sure. There's another initiative that has developed fairly recently. It's several years old now. It's called the Sweet Potato for Profit and Health Initiative. The FPHI is a multi-partner, multi-donor initiative that seeks to reduce child undernutrition and improve smallholder incomes in 10 million families by 2020. Folks, it's 2016. So we've got a big job ahead of us if we're going to reach this goal by 2020. And I can tell you that I think we're well on our way to that target at this point, and perhaps that can be a future webinar by one of our team members to describe that. Now, the lead agency for SASHA and the FPHI is the International Potato Center, based in Lima. Largely, a lot of these efforts are led out of the substation in Beka or at ILRI in Nairobi, and of course, it's led by Jan. NC State is a member of the Sweet Potato for Profit and Health Initiative, and with the support of the foundation, we have been able to develop the GT4SP project, and I'll tell you a little bit more about that in just a few minutes. So let's sit back for a moment and take what I'd call a sweet potato global status check. One, I think it's fair to say that the importance and potential of sweet potatoes is becoming widely recognized across the globe. Next week, I'll leave for a trip to China to discuss sweet potato, and literally, I have traveled around the globe several times to discuss sweet potato, and it really is an up-and-coming crop in many, many countries. So many public and private organizations recognize the superior nutritional value in sweet potato compared to many of the other staple crops, and investments are really increasing. SASHA 1, as I mentioned before, has been extremely successful. It was renewed and funded. SASHA 2 and the Sweet Potato for Profit and Health are gaining more traction all the time and gaining new partners all the time. And if you're not familiar with the Sweet Potato Knowledge Portal, I would reference you to that portal to learn more about all things sweet potato across sub-Saharan Africa and indeed the globe at this point. It's a great website for sweet potato. So in short, a lot of people are more interested in sweet potato than ever before. However, I'm a breeder, and when I survey the genetic situation and the genomic resources for improving sweet potato, they're noticeably lacking. And that's the focus of the GT4SP project, is to improve the genomic resources available for sweet potato. Now, let's just take a quick look at what's out there right now, and this is certainly not an exhaustive look, but it's just a quick and dirty look at some things that we've done here at NC State in collaboration with the International Potato Center over the years and some national agricultural research partners, namely the National Agricultural Research Organization in Uganda and at Nakri. But first, if you just do a quick Google search, which a colleague of mine, Avais Khan at SIP, has done recently, just a quick Google search on various crop search terms, for example, crop and genomic, or crop and genomic for 2014, what you're going to see is compared to the other major staple crops, wheat, rice, soybean, sugarcane, you're going to see just in terms of the number of hits when you do a quick search on Google Scholar is really low compared to many of the other major crops. Even compared to cassava and yam, it's fairly low. So we have a lot of work to do. Now, I'd like to give some credit to some of the early funders of the sweet potato genomics work, and that goes to the McKnight Foundation Collaborative Crop Research Program. Actually, my program, the University of Greenwich and the National Resources Institute, and the Namalangi, or the National Crops Resources Research Institute in Namalangi, Uganda, have been engaged in a long-term capacity development and breeding project about the past 8, well, actually, I should update this. It's not the 8 to 10 years. We've been going back all the way to 1996. So we've had a long-term relationship for 20 years now, and the focus of that effort has been to develop high-yielding, high dry matter, orange flesh cultivars with increased resistance to viruses and insects. And really the guy who got the ball started all of this is Dr. Robert Mwanga. And just last week I had the great pleasure of welcoming Robert back to our institution here at NC State, where he graduated after 15 years of being away. And I think he'd say that campus has changed remarkably, but he really got a great project started, and it persists until today. So I want to give Robert a lot of credit for that. Our focus, like I said before, was development of high-yielding multiple resistant sweet potato germplasm. We focused on the orange flesh high dry matter type, and across sub-Saharan Africa, the viruses, sweet potato virus disease, and sweet potato weevils continue to be real scourges, and they're actually going to be a major emphasis for the GT4SP project. These are still major biotic constraints to sweet potatoes in Africa. This would be anthracnose in sweet potato also, primarily a problem in the highlands of Africa, another issue that we'll be wrestling with, but really these two up here, the viruses and the weevils, if we could do something to change this and develop cultivars with improved resistance to those two pests, it would be a major step forward. So we've done a little bit of working in genetic mapping. Two people that have led that effort here would be Dr. Jim Carlos Cervantes, who's now a carrot breeder with Vilmorin, and Dr. Bernard Yada, who's now back at Nakri and leading the Ugandan sweet potato breeding program. And here's a picture of Robert some years ago in one of his polycross breeding nurseries. And then Dr. Gareta Semakula has picked up and is working in the project also. We were able, through this project, to complete the first complete genetic linkage map of sweet potato in a population Tanzania by Beauregard. And this was a great first step using amplified fragment length polymorphism. And in that work, we were able to tag QTL for various traits. And this is just one example of QTLs for, this is dry matter on the red bar, this is starch on the green bar, Beta-carotene is up here. And what we were able to confirm is a negative linkage between dry matter and beta-carotene. And these bars here with an underline on them, under them, that means the QTL is for reduced dry matter and reduced starch, and this is a locus for increased beta-carotene. And all of the applied breeders who've been working with sweet potato over the years realize that there is this negative linkage. It's hard to get high dry matter, high orange flesh varieties. I believe it's fair to say that we've now broken this linkage, but certainly it hasn't been due to the use of molecular markers. It's been to all applied genetics and lots of creative and large-scale breeding efforts, largely by our African breeders. But nevertheless, we still are not able to use molecular markers in sweet potato breeding, but we need to, and this is what our project is focused on. So we've proven the concept. We can make genetic maps in a complex hexaploid polyploid. We can tag QTL. The next step is to begin to use those to facilitate breeding. Recognizing this, the foundation realized the opportunity that existed. We had a convening at the Gates Foundation in Seattle back in June of 2013, and we understood that sweet potato is poised for significant growth in sub-Saharan Africa. But in order to fully realize the long-term potential, the foundation recognized that we needed to invest in modern breeding tools and to integrate these modern breeding tools into applied breeding efforts that are connected to improved seed systems and market-based value chains. And so what I would say is the GT4SP project is very tightly connected with ongoing investments by the foundation in the Sasha project and in the seed system project they started, and many other projects that are ongoing. We're well connected in that effort. So out of that meeting, the GT4SP project was born. It's a large project. As Mercy mentioned before, I am the lead PI, and it certainly changed my life. In terms of the scope of activities, we encompass over 20 PIs, 7 institutions, 6 countries, 4 continents, 15 time zones, 1 crop, and a $12.4 million investment. All of this is for breeding tools for sweet potato. What's our vision for this project? Our vision for marker-assisted breeding in sub-Saharan Africa includes the following. 1, in order to develop robust genomic tools for sweet potato, we need good genomic resources. That means we need a reference genome. We need new, robust SNP markers. We need advanced laboratories capable of sequencing, developing, and linked with developed and developing countries so that we can work together as a team. We need both 2X cultivated or 6X mapping populations, and we need training and test populations. With all of that, we need to develop new phenotyping, analytics, and database resources. If you look at this as a pyramid, certainly the bottom of our pyramid, and probably the most important part of it, is we need human resources and we need capacity development. And we have a lot of that built into the GT4SP project. The Sasha project and other projects have the similar built in, and that's partly what this webinar is intended to do. So we need to continue to assemble and develop a dynamic team of breeders and allied disciplines to help us. We need training in the use of traditional and genomic breeding methods. We need to work on our communication and our collaboration. And we need institutional training and capacity development. And largely this institutional training and capacity development is being led by Mercy and her team, Dr. Mark Gilon and Mercy at Becca at the SIP station in Nairobi. So this is just a cartoon, if you will, on the various aspects of the GT4SP project. We have a team that's working on establishing a sweet potato genome. I'll give you a reference genome for sweet potato. I'll give you some information on that. We have a bioinformatics group that is focused on developing the bioinformatics and the analytic tools that enable us to sort of decipher the reference genome. And we have a genotyping by sequence platform that's being initially set up here at NC State. Our long-term vision is probably that's going to be transitioned to Becca in the long term through some other investments. We have a database, bioinformatics and phenotyping team that's set up that is focused on receiving all of this data, assembling it together, and making it useful for breeders. And then we have breeding and capacity development efforts that feed back into all of these higher level components. I currently have 2 PhD students with me right now, one from Uganda and one from Ghana. They're working, they started up in August, and they are working on mapping populations that are being developed under the GT4SP project. And they're both coming from national programs, the CRI in Ghana and NARO in Uganda. And the intent is once they finish their research here, they'll go back, do their field work at their respective locations, come back here, complete their PhDs, and then return to their home countries and continue to work on sweet potato. So it's an ambitious project. There's a lot of pieces to it, but so far it's going quite well. And so let me give you a couple of updates on it. We had our first project meeting. It was our startup meeting back in San Diego in January 2015. You can see this is the core group right here. Just this past January, we had our first annual meeting, and you can see that the group has grown in strength and in numbers. We've brought together a really great team of people to bear upon sweet potato, and I'm really pleased to be working with this whole team. It's truly an honor. We had our first joint SASHA/GT4SP joint meeting in Mukono, Uganda. That was last year in June, and we're getting geared up for our second joint meeting, which will be in Nairobi this coming June also. Many of you, I think, know about it already. I want to give Ted Carey a quick shout out here if he's on the call. Ted brought along a bunch of orange sunglasses, which we all agreed to put on. And I think this just kind of is it's kind of a fun moment for us to realize that we're all looking through the same common lens, if you will, orange-fleshed sweet potatoes to improve vitamin A and nutritional status in across the continent, particularly in women and children. So we all got a good laugh out of this one. We all came home with a pair of new lenses to look through. A couple of highlights for you. You know, as a project leader, when you manage a project this big, just getting all the subcontracts worked out and sorted out is a major milestone, so I had to put that out. All of our subs are all done and negotiated, and all the funds have been allocated. We actually did our first annual report back in September. During that report, we had a total of 60 milestones in the whole results framework. 10 of our 13 milestones were completed and achieved in year one, and we're off to a really good track. We've hired a project coordinator. We have 8 new PhD scientists hired, 4 PhD graduate students, and greater than 15 support staff. We've had the meetings that I've mentioned before. We have 2 new websites, a sweet potato base website, which is our database website for the breeding database information. That's established at the Boyce Thompson Institute. And we have a project website, which is at sweetpotatogenomics.cal.ncsu.edu. We've made substantial progress on establishing a reference genome for sweet potato. I'll go over that in a couple of seconds. We have a first-generation genome browser that has been released, and it will probably be publicly available within the next, I'm thinking maybe next month to 90 days. We have both diploid mapping populations that are established between a wild relative of sweet potato, Ipomoea trifida. We have started work to make a map out of these 2 populations, and what I can say is actually that map is largely completed at this point, but we're in the process of tweezing it. We have a lot of great cultivated populations that have been developed, and we have another population which I'll call the Mwanga Diversity Panel, consisting of a set of 16 sweet potato parents crossed in a in a diallel that we'll be using for doing some long-term genomic selection work in. This is going to be a great set of materials in the future as we move forward, and that's being established. All of these populations are actually being established in tissue culture so they can be shipped across national borders. And of course, our capacity building efforts have begun now at Becca. So this is a quick look at the materials that we chose to do our reference genome from. They're two putative ancestors of cultivated Ipomoea batatas, cultivated sweet potato, Ipomoea triloba and Ipomoea trifida. This is the triloba line, and this is the trifida line, and these are the end curve plots. And basically what these plots show you is if they have a single high peak, they show that they're highly homozygous, and they would be suitable for sequencing and then reassembly, just because it's a little bit easier to reassemble a fairly homozygous line. And this I. triloba is particularly good for that purpose. The N. trifida, the I. trifida is not as good as the triloba, but it's suitable. And it's interesting that both of these lines came out of our breeding program here at NC State out of a totally different project, our ornamental sweet potato breeding program. But these will be the reference genomes for sweet potato. This is the genome assembly results done by Dr. Zhengjun Fei at Boyce Thompson Institute. And what I'd like to point out to you is that they're very good, they're high quality genomes covering roughly about 82 to, well, about 80+%, 90+% of the genomes of each one of these diploid species. The estimated genome size are down here, about 526 megabase for Ipomoea trifida, and about 495, we just call it 500 for I. triloba, so roughly about the same. We've used, this is all Illumina-based sequencing. We use packed bio long reads to reduce the Illumina-based gaps, and what we found is that in this genome here, the triloba, which was actually quite good, the Illumina-based gapping, didn't really make much more of a difference in terms of the assembly. It helped, of course, compared to the trifida here, where the assembly was improved significantly more with PacBio approach after the Illumina sequencing. So we closed those gaps with the long PacBio reads and basically have improved our assembly. This is a summary of the assembly provided by Dr. Fei. I won't go into details here, but what I would like to say is that it looks like both the trifida and the triloba are suitable for use as a reference genome for the cultivated sweet potato. And it looks like that cultivated sweet potato probably consisted of two ancestral genomes, one a tetraploid trifida, and second, a triloba. So this would be the predominant contributor to cultivated sweet potato, and triloba lesser so, but we're still exploring that hypothesis, and this would be a first out of this project. Dr. Fei sent all of the information to Dr. Robin Buell, who is now putting together a genome browser. We have an estimated number of genes of roughly about 34,000 genes in both species. This is the average gene length and some of the metrics of the genome. We are beginning to functionally annotate those reference genomes using various strategies. And we've developed a nomenclature for each one of the locuses. And this has all been implemented in a JBrowse genome browser. This would be the first genome browser for sweet potato. Again, another first for genomic references for sweet potato. We will be doing training for this in due course. Truth be told, as an applied breeder, this to me is a bit braille, but basically it's a great roadmap to identify genes, use those identified genes as potential candidate markers, candidate genes in mapping populations. So it is a very good resource as we're moving forward for sweet potato to understand the complexity of sweet potato, understand the genetic makeup, where genes are located, how they're structured, et cetera, that are going to be very useful moving forward. And we'll be through probably webinars doing some advanced training on how an applied breeder might begin to look at and utilize a genome browser in their era of genomic breeding. I mentioned earlier that at NC State we're developing a genotyping by sequencing platform. That's coming along quite well. Our strategy is to develop both low and high density genome-wide SNP pipelines. It will be at multiple plex, and we assume that we're going to have uniform coverage of loci and alleles across the entire genome. We see that there are going to be some areas we're going to have high coverage so that we can call dosage in the complex sweet potato polyploid. In certain cases, we might not use imputation, but in many cases, since we have a reference genome, we may be well trying to imputate calls based on our knowledge of the reference genome, all right? And we first need to determine that, and we're well on the way to determining that, what's going to be the best reference genome for calling SNPs in hexaploid sweet potato. A little bit more strategy, we want to develop both low density SNP maps and high density maps for different breeding purposes, and we're going to make sure that these are reasonably cost effective. They'll exclude repeat reasons, exclude unusable SNPs, and we'll use methylation sensitive enzymes similar to the Cornell protocol, except we're doing a few things in terms of sizing and optimizing our enzymes for sweet potatoes. This is just a quick flow diagram of that. I won't go into the details of it right here, but it really is the standard GBS protocol for sweet potato. The only thing that we're really changing is we're changing the way we're developing our barcodes. We're using different double digest enzymes, changing our barcodes a little bit. We're size selecting, and then we're doing the sequencing, and we're working on that right here. We had the team here last week that's managing the phenotypic data with the genotypic data, led by Dr. Lucas Mueller's group at Boyce Thompson Institute. This effort is now coming along right now, and we'll be instituting some training programs in that. Again, I think a webinar methodology is a very good methodology to do this, but we're also going to be doing a lot of face-to-face training at the individual breeding programs as time progresses. This is just a quick look at sweet potato base right now. It has been constructed. We have not yet populated it with much data, and that's going to be the next step, is to really begin to populate this database with actual mapping experiments that we're engaged in, and to have the breeding programs begin to use the database as a tool in their breeding program. This, again, is a radical departure from the past, and so there's going to be a lot of training that's going to be required for that. This is just a picture of the team that was here last week. It's the database group right here, standing in front of what we call the Tally Center at NC State. We took a day off to tour the field. Sometimes it's good that we had a mixture of actual breeders in the group, then bioinformaticists in the group. This is Lucas Mueller right over here, leads the database group. Geneticist Avais Khan, Bernard Yada, Robert Wonga back, they're alumni, genomicists. So we took the time to get them out in the field to see how we do it in North Carolina. And for those of you that are unfamiliar with our planting process in North Carolina, these are actually sweet potatoes right here that are being laid out in a row in a process called bedding. This cart actually is delivering the sweet potatoes, laying them out on the row right now. These are going to be covered with soil in the next, these will be covered with soil. The next step in this planting operation will be cover them with soil, and then sprouts will emerge from these roots right here. And then in North Carolina, what we do is we cut the sprouts that emerge from the roots and transfer those into the commercial fields. Very different from what we do in sub-Saharan Africa. Okay, so I'm going to wind down here, and here's a couple of things that I've learned from lessons that I've learned from year one. One, I've learned how critical it is for a reference genome. And once you have a reference genome for sweet potato, how important it will be. I understand that we need a sequence-based genotyping platform. What that will ultimately look like is yet to be determined, but we're building the flexibility into our projects so that we can understand that. We need breeder-friendly bioinformatics and analytical environments. And when I say something like this is very easier said than done. What it will look like, what are the special needs for clonally propagated polyploid crops, and how that's going to look. And I also know that we're going to need technical support for breeders. This is a big change for us in here, and this is going to require a lot of training and a lot of hand-holding. We also need good populations to work with, but I'm not really worried about that because we have some great breeders that are engaged with the program, and we're going to be able to develop as many populations as we need. What's really going to be the trick is to incorporate these aspects into our breeding work and incorporate the genome aspects into the breeding work too. If you're doing all of this and you're doing more mapping, we need improved phenotyping and data collection capabilities. We also need mechanization. We simply can't handle the number of progenies by hand anymore. We're going to need sustained funding, and that sustained funding is going to have to come from multiple sources, not just one entity. So we as a community are going to have to work on that together. And I dare say that it's not only going to be the sweet potato community that works together to do this. It's going to be the sweet potato, the yam, the cassava, all of those other communities working together. We will need sustained capacity development, and this is critical. So what I'd like to say is just leave you with those concluding thoughts. And what I would say is our project is a very big project. This is a list of the institutions and the lead PIs for each group. It's a great team, and it's a real honor to work with each and every one of them. And as an applied breeder, feel terribly fortunate that I can work with such a diverse range of people, and I think it's emblematic of what breeders do nowadays. Breeders don't only work with breeders now. You work with bioinformaticists, you work with programmers, you work with food scientists, you work with pathologists, you work with entomologists. It really is an exciting discipline, and it's a fun job, and I feel fortunate to be engaged in it. So with that, I'll leave, and that's my last slide, I believe. And if I have time, what I would say is stay tuned and stay engaged. And I hope that this seminar has helped you get a slightly better idea of what the GT4SP project is all about, and certainly what sweet potato is all about across the globe. Thank you. Mercy, it's back to you.
The genomic tools we're going to develop. And so from the audience, I'll take your questions. From the list of questions that were sent on the chat, I have one question. And I think I should open the floor for the questions. And then, because I haven't gotten your questions on the chat, you may have to ask from where you are. We still have about 7 minutes to go to finish our webinar, and Mark and Craig will answer our questions.
I'll do my best. And
Craig,
go ahead.
Yeah, Craig and Mark, when you are presenting, you mentioned quite a number of traits that the project is focusing on. And from a breeder's perspective, my question is, are you going to focus on the improvement of all these traits? And you mentioned high dry matter, orange flesh, high yield, resistant to diseases and pests. And if you're going to focus on all of these traits simultaneously, what advantage do we have with the breeding tool that the project is developing. That is the reference genome and the mapping population. What advantage do we have? We have the reference genome and the mapping population. Thank
you. Yeah. The short answer is yes, we are going to focus on all those traits as breeders, but I don't think that we'll use genomic tools to facilitate breeding for all those traits. And perhaps my first two major targets for a reasonable marker-assisted breeding platform in sweet potato would probably be sweet potato virus disease and perhaps sweet potato weevil, if we can identify loci that are associated with those two traits. And my reason for saying that is because they are such difficult traits to breed for already. And if we had a system, a marker or markers that could help us identify progeny early on in the breeding process and advance those, that would be much more beneficial to us. So I think we have to be prudent in trying to determine, what we can and what we can't do. It's yet to be determined right now how the reference genome will help us, but there's plenty of opportunities for known genes for understanding if we have a QTL by looking for closely located SNPs or SNPs that are in candidate genes that we can understand the physiological or physiochemical basis of a trait. So that's where the reference genome is going to come in handy for something like that. In the long term, there's this emerging concept called genomic selection, where you can identify the breeding value of a particular clone or set of clones and use those to make your optimal crosses. And I view that the Mwanga diversity panel is probably going to allow us to test those hypotheses in sweet potato. So we'll be looking at that also. But my feeling is right now is first we need to make the tools and we need to apply them to simple targets and then build upon that knowledge.
Okay, thanks, Craig. There's one more question, and please, if you have a question, you can raise your hand, and then I'll see you and call you to ask the question. There's another question of how long has it taken to get the reference genome of the sweet potato up to this point?
Actually, that has come remarkably quick, and it's because of a unique confluence of beneficial, just some great confluence of good circumstances. Probably the reference genome has been developed in the space of 1.5 years, and the genome browser will be posted probably in the next 60 days. And then it's going to be an iterative process where we enhance that browser with improved resources and the community feeding back on that and annotating that our reference genome through the browser. So version 2 is going to be out pretty soon, and that'll be a first. And then it's going to be an iterative process where we'll slowly improve it. But it's really come a lot quicker than I had expected. And I think it's because we had the right germplasm in place, we have new sequencing technologies, and we have a stellar team that's implementing it.
Okay, thank you, Craig. More questions from the audience?
I see somebody raised their hand before. Ahmad, was it?
There's Muhammad Ali. Muhammad Ali, could you please ask your question?
Yes, I'm master's student in crop economics in China and basically from Pakistan. So I'm asking.
You speak louder.
I can't hear.
I also can't hear. Muhammad Ali.
Yes, I'm from Pakistan. I'm studying crop economics in China. I'm a scholarship student. So I'm asking that you are working in North Carolina State University, and you are working with sweet potato, which is basically grown in Africa and some Asian countries like China. So how we can get benefits from assemblies and governments, and other what we have done in the development of sweet potato. How the breeder can basically get benefits from the work you do in the nursery. Breeders mostly they are in Africa, India, and they have no facilities to do genomic research. So how we can get benefits from the work we have done?
Sorry, Hamad, we did not get your question clearly. Could you please use the chat and type your question?
Yes, that's a good idea.
Most of the breeders are working in the less developed or underdeveloped countries. Can we just rely on the community? I'm asking them how do we get benefits from the work we have done?
Okay, Hamad, I've sent you a text. Kindly type your question because we can't hear you clearly.
Okay, I'm typing.
The next question.
He should close his mic.
Bramwell, you have a question? Hamad, please close your microphone and state your question. We'll take it back. Bramwell? Bramwell Wanjala, kindly ask your question. Your hand was raised up. Craig?
Yes.
Craig, can you hear me? Yeah.
Yes, I can.
Hamad will be texting or typing her question. And I don't know if there's anybody in the audience with more questions. Kindly ask, raise your hand. I'll see it and you'll have a chance to ask the question.
Okay, gotcha. Yeah, I'm not seeing the host.
Okay.
Yeah.
Seems like we've got no more questions from the audience. Great.
Yep.
Okay. And so to the audience will be, as I said when we were starting, this is the first of a series of webinars that will be, the Genomic Tools Project will be organizing to educate and inform the sweet potato breeders and the wider communities of practice who are working on other crops with complicated or complex genetics. So as Craig put it, please stay tuned. We'll be sending an invitation to the next webinar.
I'm still here.
Yeah. Does anyone have a question? Julius Terumanga?
Can you hear me?
Yes, I can hear you.
Yeah.
Please go ahead.
Okay. Thanks for the invitation. And my question is, or clarification, tremendous progress has been done through conventional breeding, whereby there has been a lot of population improvement towards different stresses. And I want to know what plans they have to characterize the local adapted germplasm, and also with the reference to the mapping population, what do they know about the positions of these criteria in different backgrounds?
It's a very good question. One, there's been a lot of work done in many of the local breeding programs in sweet potato to characterize the local germplasm. But I think with the Genomic Tools Project, we're going to come up with a set of easy-to-use markers in the future and some protocols that can be implemented more rapidly in collaboration with, say, Becca or with the genotyping platform. So my vision would be is that we have a good pipeline. Maybe one or two of your students that might be working on that would do that locally, and we'd offer a service in terms of characterizing diversity. So I think that's an opportunity right there. In terms of the mapping populations, we're developing new mapping populations, but we have a very long way to go to actually use those. And so I think that for me, the analogy that would be was we're building a football field, if you will. And when we build that football field, we want more and more players to come play on it. But first, we need the stadium. So we're building the stadium, and then we have other people who can come and use that stadium, if you will.
Thank you.
Did that answer your question? I mean,
Julius, was your question answered?
Yeah, the question is clear that this is just the foundation, and there are yet more things to be done on the characterization of the germplasm we have. Thank you.
Yeah, that's very true, Julius. Yeah.
Okay, yeah. Thanks, Julius, for the question. It's very valid. And Ahmed sent her question, and our question, Craig, you can hear me. Our question was how can the breeders in the less developed countries get benefits from the work that the Genomic Tools Project is doing so far? And because as we, the breeders, are mostly from less developed countries. Yeah. So how will the breeders from the less developed countries who are working on sweet potato benefit from this project?
Mercy, I'm going to let you answer that question as our capacity development person, and I think you have a very good knowledge of that, and you can signal that from our perspective.
Okay. So, Hamad, can you hear me? Thanks, Craig. Ali? Yes, I know you can hear me. Okay, now this is how you benefit. Craig mentions that one of our objectives is to do capacity building, and this capacity building we are doing in Sub-Saharan Africa, and these are in countries which are less developed. And now what we've started doing is by getting people to come, because we are in collaboration with Becca, or the Biosciences Eastern and Central Africa, to get breeders from those less developed countries to bring them, train them in Becca, and teach them how they can use the marker-assisted breeding to improve their sweet potato breeding program. So for last year, a call went, and one of the avenues we are using is the African Biosciences Challenge Fund, which is a fund which is funded by the Bill and Melinda Gates. And so we've gotten a few candidates to come here at Becca for about a month, 3 months to 1 year, to come and train on molecular breeding. The other avenues we are using is through workshops and also through consultations. Like if you go to the Sweet Potato Knowledge Portal or to the Genomic Tools Project website, we'll be posting a number of items that you can utilize or learn and choose them in your breeding program.
Yeah, I'd like to I'd like to just kind of follow up on that a little bit too, is because I'm very sensitive to this, and I recognize the fact that most developing countries, particularly some developing countries in sub-Saharan Africa, really don't have access to these high-powered resources, and very much the way that I view it is sort of like Mercy described. We'll have training and capacity development, but what I want to see and what we've seen through the SASHA project is a community of practices developing, and I would almost view them as overlapping circles, in a Venn diagram. And so it might be that you can reach out to Becca, and you can send some of your technicians to Becca for training, or say Mercy can go there and give you some training. And then they might be able to access NC State. So it would be that chain phenomenon, and we just build that capacity slowly. And I think it's important to know is my horizon is not, the next 2 years. It's the next decade. We're talking breeding time. And my whole philosophy has been is we need to begin the process. We just need to begin the journey together and then to begin to sensitize people. But I don't claim that this is going to be a revolutionary change to your breeding program, say in Zambia or in Malawi or so, in the next 5 years. But we really do have to change it. And over time, we will increase our capacity together to do things. And the technology is going to change rapidly during that time, too. So it's a long-term process. I understand that.
Okay. Thank you, Craig. Muhammad, I have your email. I can get back to you with all this information on how you can, what the genomic tool or how the genomic project can help you to access these facilities. And we are.
Being an experienced scientist, I want to contribute something to the betterment of the African people. But unfortunately, I have no resources to do anything for them. But just I can do my suggestion, and anything I can do, I will do. And when I graduate from my master, and I want to do my which are basically cultivated in the Africa, those are indigenous to Africa. Thank you.
Okay, Hamad, I'll be sending you an email with all this information, and then we can pick it up from there. Thank you. Okay. Thank you very much, Craig, and all the participants today for today's webinar. And I am delighted to have you all participating. And stay tuned and look forward to our next webinar, which will be advertised soon. Thank you very much.
Thanks for hosting, Mercy. All right.
Yes. Bye.
Thank you.
Bye-bye, everybody.